Hello, everyone. I hope you all are doing well. My name is Olivia Bedoin, and I'm here to talk to you all about a species of cephalopod, Octopus vulgaris, or the common octopus. These organisms are a great study species for the important role they play as a predator in reef communities and beyond. They are also a topic of interest for their well-developed cognitive abilities, but for this research done by Smith, we are going to be focusing on predator-prey interactions. So to give you all a bit of a background information on the species, octopi are of the phylum Mollusca, class Cephalopoda. They occur in tropical, subtropical, and temperate waters of the East Atlantic Ocean and Mediterranean Sea. Although capable of a wide array of cognitive abilities, such as short-term and long-term memory, mimicry, and problem-solving, one cognitive function that can be explored is their foraging capabilities. Octopus vulgaris are specializing generalists, which means that they are flexible to the type of prey available, but prefer to consume what is readily available. This species is of importance for study because of their ecological role as a generalist predator in community interactions and the large role they play in commercial fisheries in South Africa. Some factors that have previously been recognized as influencers on their diet are prey abundance, depth, habitat type, and maturity of the octopus. They are also a notable species to study in South Africa because at times populations can be exploited by fisheries, which could not only impact the population of octopi itself, but the species also interconnected within their food web. So in a wide ecological lens, the topic of this research focuses on species interactions between Octopus vulgaris and its prey. More specifically, what type of diet does the population of octopus in False Bay, South Africa, have as a result of their individual size? In general, they are looking to see if there's a correlation between the size of the individual octopus and a specific diet they consume. Overall, this research explores what types of prey the population of octopi consume and the ratios of the prey in their diet. So in other words, what prey has the most impact on the octopi and vice versa? What prey do octopi have the most impact on? To give you all a better picture of where this study was conducted, here is a geographical representation of False Bay, South Africa itself. In Figure 1, you can see Cape Town in the lower left-hand corner of the map, which is one of the most popular tourist cities in South Africa. Right below is a little hook that encloses False Bay. Figure 2 shows a satellite image of False Bay where we can see this enclosed area of the ocean where a lot of action could potentially be going on. Taking an even closer look, here's an annotated image of the study sites. We have Glencairn, Windmill Beach, Miller's Point, Buffalo's Bay, and Cape Hangcliffe. Separate sites were chosen for a wider geographical range of data, so we know that the data found isn't occurring in only one part of the bay. These sites were also chosen for their kelp beds and reefs as good habitats for the octopi in their communities. The team used 3 methods for getting a better understanding of octopus diet. First, octopi are captured and brought back to the lab for stomach content examination. Here, they dissect the individual and note any important taxa found within the food pieces in their stomach. Next, after the octopi were removed from their dens, midden piles, which are their leftovers from their meals, were taken back to the lab and also examined to pick out any species they found. Lastly, direct observation was made with dives, noting any observations of prey capture and consumption. For a more detailed look at the methods, sampling was done from February 1997 to January 1998. Again, these 3 methods included capture and dissection, collection of middens and scrutinization of them, and instantaneous observations. It is important to note that they grouped any middens as an instantaneous observation if they still had flesh on them. Furthermore, abalone shells were used as a baseline for prey size selectivity because most samples were intact. This is crucial for comparing octopus size with the size of prey consumed, specifically looking at the abalones. Lastly, in Figure 4, this is a representation of what an octopus den looks like. You can see the homeowner is still occupying the den, and in the lower left-hand corner of the den, these are what the middens look like. Next, it is key to consider some factors that contribute to the overall strength of the research and factors that could be considered confounding. The researchers noted sampling techniques could have contributed to the results. For example, night versus day with field observations could play a role, as only daytime observations were recorded. So the octopi could have different hunting techniques at night, or there could be different prey available at night. A strength of the field observations is that they are useful for determining if the species found in the dens are actually consumed, and this also is supported with stomach content analysis. Overall, the researchers agreed that the stomach contents was the best method for collection because it shows what prey is actually being consumed in a recent period of time, and the hard-shelled taxa can easily be identified because they are not digested right away, meaning they would have a larger gut retention time. Conversely, I would also argue this is a weakness because soft-bodied creatures in the stomach would then likely be digested quicker. But stomach contents can determine the importance of small prey items in their diet. Furthermore, midden counts can easily be disturbed by biotic and abiotic factors, such as being swept away by currents or any degradation within the den. Lastly, midden collection is useful for easy identification of prey, and the prey size can be measured. So, a major result that they found was that prey size and diversity increased with increasing octopus size. In total, 336 octopi were collected, and the majority of which were found resting in their den. 39 prey species were identified, and most of the identification came from the middens. Of these 39 prey species, the species that had the most significant role in the overall diet of the octopus population were Plagusia charabras, the red bait crab pictured in Figure 5, and Haliotis midday, the South African abalone pictured in Figure 6. Continuing with what they found, a high majority of the prey in the stomach contents were crustaceans or soft-bodied organisms. The middens had fewer crustaceans and soft-bodied organisms while having more shelled mollusks. Field observations noted a high amount, 63% of abalones, which could be contributed to a small sample size, but there were significant differences in the mean size of abalone consumed by differing sizes of octopi. Small octopi consumed abalones of 53.3 millimeters, medium octopi consumed abalones of 72.6 millimeters, and large octopi consumed abalones of 86 millimeters. In total, the frequency of occurrence in the diet is held 63.6% by crustaceans, 37.6% by mollusks, 11.2% by telosts, and 10.8% by polychaetes. Before I show you representational figures of the data, I just wanted to summarize how it was presented. Each prey item in this pie chart correlates with a textured section of it. The species with the largest importance on the diet occupy the largest portion of the chart, such as the small crustaceans in this example figure. I also wanted to point out that the overall importance of prey species in the diet of the octopus was assessed with the index of relative importance. This is measured by taking the percent of the number of prey species in the gut added to the volume of the prey species in the gut, which was then multiplied by the frequency of occurrence in the prey species in the gut. So in sum, Figure 8 represents the three separate charts that correlate with the instantaneous daytime observations, the midden counts, and stomach contents. We can see with the field observations at the top that abalones were recorded the most. Midden collections were primarily composed of abalones, winkles, and limpets in that order. The stomach contents were primarily composed of large crustaceans, small crustaceans, and abalones. Over on the left, Figure 9 displays each octopus size, small, medium, and large, and the diet that correlates with them. The small octopus had a diet of mostly small crustaceans, large crustaceans, and winkles. Next, the medium octopus had a diet of mostly large crustaceans, small crustaceans, and abalones. Lastly, the large octopus had a diet consisting mostly of large crustaceans, small crustaceans, and abalones as well. And right here is another figure showing the size comparison of octopi with their preference of size and prey, specifically with the abalones. As seen with the trends on the histograms, there is a greater abundance of larger abalones paired with larger octopi, a greater abundance of medium-sized abalones paired with medium-sized octopi, in smaller abalones paired with smaller octopi. We can also see that the larger octopi consume a wider variety of abalone size than the smaller octopi. So, what do these results mean for this field of research and beyond? In general, this research provides a closer look at the predator and prey interactions of this area, and this is vital to see how prey abundance can directly impact octopus feeding methods. On the flip side, this gives us a look at how octopi feeding methods or abundance can potentially have a future impact on prey abundance. Even further, the size of the individual octopus matters in this assessment. This displays more evidence towards the opportunistic feeding habits of octopi. Although different sites were used for a wider geographical range of data, no major differences in results were mentioned between sites. Locally, abalone populations are declining, and they were shown to be a huge component in their diet, which means that this assessment is crucial for the management strategies of fishing on abalones and octopi themselves. Aside from creating management strategies, this study is another step in the continuation of analyzing community interactions of octopi and their cohorts. Some questions that arose from this research that could be studied in the future are: how does seasonality affect the diet of Octopus vulgaris in False Bay? Does the presence of competitors affect the diet of Octopus vulgaris in False Bay? How does the diet composition of Octopus vulgaris change when abalones are removed as an option, which could potentially happen in the future if this population is declining? Lastly, I'd like to again acknowledge Smith and this great piece of research done. It is an insight into the life of an octopus in this area and is a great assessment of their foraging behavior. And I wanted to thank you all for your time and hope you have a lovely day.